Hello students, welcome to Royal Palace PU College Jamkhandi online classes. We already studied some of the basic videos relating to a variety of chapters now. Now what I have done, I have prepared one video consisting of the explanations of the entire chapter of chapter number 10. The name of that chapter is second year PUC, chapter number 10, role of microbes, right? Microbes in our day-to-day life, the use of microbes in our day-to-day life, what is the application of these microbes in our lifestyle, that is microbes in human welfare. Dear students, watch this video in one short video which gives you an entire explanation of the chapter, which I have made it short into less than 1 hour only. Right? It completes the entire chapter. So be aware of this chapter. Watch this easily. Go with easily. Try to read the sentences, each and every sentences. It will give you an idea. Okay, students, let's get started about this chapter.
Today we'll be starting a new chapter of second PUC biology, and that is microbes in human welfare. And the today's topic is role of microbes in household products. Dear students, since we know that there are different types of microbes including bacteria, virus, fungi, etc., etc., we cannot say that all the bacteria, all the virus and all the fungi are disease causing in nature or harmful in nature. We can list a very variety of different types of bacteria and fungi which are beneficial to human beings. So this is the chapter which tells us that what is the actual role of different types of bacteria and fungus in our day-to-day life. And today's topic, as I mentioned, in our day-to-day household activities and in products. In this, I'll be giving you some 4 to 5 examples which explains us very clearly that these bacteria and fungus are needed to us. The first example is curd. Curd is prepared by adding a small amount of curd as a starter to the milk. The milk contains casein as a protein, and it contains a specialized group of bacteria called as lactobacillus or lactic acid bacteria, which on a suitable temperature converts that particular milk protein into the curd, which we say that the coagulation of the milk. We have to remember the key microbe used here is a bacteria which is called as lactic acid bacteria or lactobacillus or LAB. We even have some benefits of LAB. It increases vitamin B12 concentration, thus increasing the nutrient content of the milk. It even detects or checks some of the disease-causing pathogens present in our gut. The second example is dough. Dough is prepared by the fermentation of the bacteria. In the process of making bread, we use a fungus called as Saccharomyces cerevisiae, which is also called as baker's yeast. And this Saccharomyces cerevisiae, upon fermentation, produces the bread. We have seen a puffy appearance to the dough which is being created in the process of making bakery products, and that puffy appearance is because of the release of carbon dioxide which is produced during that time. We can even go for some more fermentation products wherein which the carbon dioxide is also released and the baker's yeast Saccharomyces cerevisiae is also used, such as idli, dosa, etc., etc. The third example is toddy. It is actually used as a traditional drink in southern India. It is produced by the fermentation of sap which is produced from the palm trees, and this fermentation is done by a bacteria. The next example is cheese. Cheese is formed by the partial degradation of milk by the help of different types of microorganisms. The first example in cheese is Swiss cheese. We have seen this Swiss cheese by the presence of its large holes because of the production of different or large quantity of carbon dioxide in it. The Swiss cheese is produced by a bacterium called as Propionibacterium shermani. Once again, let me tell you that this Swiss cheese is identifiable by its color, that is yellow in nature, and presence of large holes created by the large content of carbon dioxide released during the process. The next cheese is called as Roquefort cheese. It is also created by a fungus called as Penicillium roqueforti. Its character is that it contains a specialized characteristic odor or smell in it. The microbes are also used to ferment fish, soybean and bamboo shoots to make foods. So dear students, these are some of the basic examples which we can study in this particular topic of the chapter. We can list a lot of examples relating to the role of microbes in household products. But since we are limited to our NCERT syllabus, or that is the only thing which is important to us, we need to remember these 4 to 5 examples. The first example is curd, the second one is dough, the third one is toddy, and the fourth one is cheese. We need to remember what are the different microbes. Is it a bacteria or a fungus which is used by the partial coagulation or from the fermentation work. So in the NEET examination and even in the theory examinations also, these are the basic questions that are the basic points that we need to remember. So students, this is the end of this topic, and today we'll be starting a new topic of the previous chapter called as the role of microbes in human welfare, and today's topic is role of microbes in industrial products. In this particular topic, we'll be studying 5 different sections and the use of microbes in these particular industries or sections which produces products helpful to humankind. Actually, when I say about industrial products, it should follow some processes such as fermentations, which is nothing but the process of conversion of glucose into organic acids or the alcohol by using large vessels or containers called as fermenters. The first section in industrial products is fermented beverages. The beverages are formed by the fermentation process of malted cereals. Now the question is, what is the meaning of malt? We'll be taking cereals or grains, we'll add them in water. Inside the water, after some period of time, they sprout or they germinate. After the germination, we halt the process or pause the process and we allow it to dry by using hot air, and after some time we'll powder it, we'll add some more water, and then we'll add an industrial microbe such as Saccharomyces cerevisiae or brewer's yeast. The yeast starts eating all these germinated, sprouted cereals and converts that glucose into alcohol or other its derivatives. This is called as fermentation process, and from this we can produce a lot of beverages. Now all the cereals or all the grains or all the fruit juices does not have the same kind of fermentation process. It depends upon different grains and different processing methodologies. Some methodologies undergo with distillation and some without distillation. I can give you some examples of with distillation such as whiskey, brandy and rum. These are the alcoholic beverages which are produced compulsorily with the process of distillation. And some examples such as wine and beer are produced without the help of distillation. The second category is antibiotics. The word itself indicates that anti means against, bios means life. Now the meaning does not compulsorily say that it is against the life of human individual. No, it is actually against the life of the particular pathogen which is causing the disease. These are the chemicals or the compounds which are produced by the microorganisms itself to kill those microorganisms or the related microorganisms. It may be a fungus or it may be a bacteria. In one of my classes, I'll be explaining what actually is the antibiotic, how it is produced. I can define antibiotic as these are the chemical substances which are produced or manufactured by the microorganisms itself to kill or to stop or to retard the growth of the pathogens. We need to remember the first created or produced or discovered antibiotic here. The name of the antibiotic is penicillin and was discovered by Alexander Fleming. And it was actually an accidental discovery by him during the World War II. Actually, he was working on some bacterial strains, and while working he found out that there is a particular category of microbes which will not allow the growth of other bacteria, and he found out this particular fungus called as Penicillium notatum, and from that he isolated this particular antibiotic called as penicillin. But this wasn't exposed until and unless there were two scientists who showed the actual work of the penicillin, and they are Ernst Chain and Howard Florey. All these three scientists, such as the Alexander Fleming, Chain and Florey, awarded by the Nobel Prize. We can even add some of the diseases which are cured and which can be cured by the antibiotics, such as plague, whooping cough or kali khasi, diphtheria or galghotu, and leprosy or kushtarog. The third category is organic acids and alcohol. Whenever we use a word called as organic acid or organic, we should have to understand that these are the acids which are derived or which are including some of the basic elements of life, such as carbon, hydrogen, oxygen and nitrogen, or we simply call them as corn. In industries, with the help of microbial activity, there is a production of large section of organic acids. I can list some of the organic acids which are given in our NCERT. The table gives us a clear understanding wherein which I have given the first content as organic acid, the second one as the microbe from which it is produced, and the third one the type of microbe. The first example is citric acid. It is produced by a fungi called as Aspergillus niger or niger. The second one is acetic acid, which is produced by a bacteria called as Acetobacter aceti. The third one is butyric acid, which is created by a bacteria called as Clostridium butylicum. The fourth one is lactic acid, produced by a bacteria called as Lactobacillus and its related species. We can even take the example of ethanol or ethyl alcohol, which is produced by using Saccharomyces cerevisiae as a basic substrate product. The next section is enzymes. As we all know that enzymes are the substances, chemical substances or biocatalysts or chemical catalysts which increases the rate of a reaction without involving in the reaction. In our NCERT relating to this topic, we have got some 2-3 examples of enzymes and their uses in different industrial works. Here I have given 2 columns containing a type of enzyme and its uses. The first section is lipases. These are the lipid derivatives, and the name itself indicates that these are used to remove the lipid content or fat content or oily content or grease content. These are used in the detergent formulations, and these are heavily used in removal of the oily stains from the laundries. The second section is pectinases and proteases. These are the protein derivatives, and these are used in the clarifying bottled juices to maintain their lifespan, to maintain their life cycle assessment for the longer period of time. The third content is called as streptokinase. This is also an enzyme formed by the carbohydrate derivative, and it is produced by the streptococcus, and it is modified by the genetic engineering. Generally, this streptokinase is used in case of medical treatments or medical industries as a clot buster in some of the operations such as myocardial infarctions related or heart related. After the operation, there is a possibility of formation of clots in the blood vessels. Streptokinase is an enzyme which helps in the removal of these particular clots, hence the name clot buster. The next section is bioactive molecules. The name itself indicates that these are active molecules which are helpful for the humankind in many respects. These are not at all used for the longer period of time. These are used only for some certain operations or some temporary period of time. The first molecule is cyclosporin A, and this is produced by a source organism called as fungus. The name of this fungus is Trichoderma polysporum. Generally, this cyclosporin A is used as immunosuppressive agent in organ transplant patients. In case of organ transplantation, let us take an example called as the heart. We have transplanted a heart. Now this heart, according to the brain and the subordinates of the brain, thinks that this is an antigen. This is not at all acceptable to our body. So we need to force it and we need to reject it. Although the body's compatibility with that foreign heart is more than 90% similar. During these cases, for a particular period of time, our immune response should be deactivated. This process is called as suppression of immune activity. To do this, we have got a chemical called as cyclosporin A. This chemical binds with specific receptors which enter into the signaling process of the brain and stop the activity of brain. The next section is statin. Statin, it is produced by a fungus called as Monascus purpureus. The help of statin is that it is used as blood cholesterol lowering agent in our body. In many of the obese people or many of the oil drinking and oil eating people, wherein which oil is the main part for their diet, they eat a lot of fat. During that time, it is a possibility that their blood vessels might fill up with a lot of cholesterol, which leads to heart attacks and some other diseases. Now, statin is a chemical bioactive molecule which helps in the lowering of blood cholesterol content of their body. So, dear students, these are the different 5 sections of industrial products which utilizes the role of microbes. Feel free to pause the video in between so that you can understand the details of it. I have given so many examples, different microbes, fungus, bacteria, etc. and their uses.
Today's chapter is the same old one, that is chapter number 10, second PUC, the role of microbes in human welfare, and today's topic is the role of microbes in sewage treatment. First of all, what is the meaning of sewage? It can be from the industries, it can be a domestic one, it can be from agriculture, a lot of things. But right now we are concentrating sewage as the municipality waste material which is collected from a lot of domestic canals or pipes, which generally contains huge amount of human excreta which can be filtered out. And what is the meaning of sewage treatment here? Treatment, we need to understand that sewage is the waste content and treatment is the purification of that. Sewage is the wastewater or dirtiest water and we need to make that dirty water into the clean, utilizable, soft or fresh water. I'm not saying the water will be 100% fresh and accurate. I'm just saying that it will be easier for the human being to utilize it for some other activities. So once again I'm telling you, welcome to the topic called as the role of microbes in the sewage treatment. The first question is, what is the need for this sewage treatment? Let me tell you, the chemicals or the contents or the liquid or the water or the dirty content which comes out of the domestic waste, it contains good amount of organic matter, and along with that even the pathogenic microbes also. We need to remove this organic matter and the pathogenic microorganisms and to make it less polluted in nature before discharging into the natural bodies. The sewage treatment is done in STPs. What is an STP? STP is nothing but sewage treatment plants. The government of India started Ganga action plan and Yamuna action plan just to remove the destructive, polluted, toxic contents of these rivers because of a lot of human activities going on there. And it's been started from a lot of years. A lot of amount has been added in this section just to make sure the water of Ganga and Yamuna are clearer for the future purposes. Now let us enter into this topic, that is the sewage treatment. Remember, we are studying not only just as a sewage treatment, we'll be studying the role of microbes in these sewage treatments. Generally, the cleaning of the wastewater, the purification of the wastewater takes place in two methodologies or two stages. And these two stages are primary treatment and the secondary treatment. Primary treatment is also called as the physical treatment, and secondary treatment is also called as chemical treatment or biological treatment. So let us jump into this topic called as primary treatment and try to understand what actually is present here. First thing, as a primary treatment, it is nothing but the physical removal of small and large particles which were present in the sewage. A clearer understanding, the primary treatment of the sewage is nothing but the removal of large particles and small particles from the sewage. A simple one. Here, the floating waste called as the debris, remember the word debris, D-E-B-R-I-S, is removed by the sequential filtration, step by step filtration, by allowing that particular wastewater or sewage to move through different filtration methods such as the screen meshes or mesh screens. This sewage is allowed to pass through these different diameter sized meshes so that the large particles along with that the small particles, or depending upon the size of the particles, they'll be filtered out. After this, we'll get a word called as grit. Grit is nothing but small pebbles and the soil. We already know what is the meaning of soil, and as far as pebbles are concerned, these are nothing but smaller stone-like structures, and these are allowed to enter into settling tanks, wherein which with respect to sedimentation or depending upon the gravity with respect to their weight, they'll allow to settle down, and after the settlement they will be removed out. Now at this stage we'll be getting two layers, the one which is floating on top of it and the one which is settled down. The one which is floating on top of it is also called as the supernatant, and here we give a word called as effluent, and the one which has already got sedimented down is called as the sludge. We give a name called as primary sludge. So sludge and primary sludge, there is no difference at all. Try to understand it. Now this primary sludge will be removed out because we are not interested in it, and the effluent or the supernatant or the one which was floating on top of it will be allowed to enter into the secondary treatment or chemical treatment or biological treatment. Now we'll try to understand what is the meaning of biological treatment or the secondary treatment. Now the first stage of the chemical treatment or the biological treatment or the secondary treatment is that We have already collected the primary effluent, and this effluent is allowed to enter into the aeration tank. Aeration tanks are nothing but general tanks or the large container which can hold up to a lot of effluent in it, and whose lid is open or open to the atmosphere. In this place, even the constant agitators or constant agitation is carried out. Agitation is nothing but these are propeller-like structures which are used for uniform mixing of that particular effluent. Remember, these are called as aeration tanks because the air is allowed to move, or the sewage is in contact with the air. That's why the name aeration tank. And since this effluent contains the organic matter and even the pathogenic microbes, and it is open to the atmosphere, it is a common understanding that there is a possibility of the useful aerobic bacteria and fungus might open, might germinate, and might grow in this particular place. While growing, these bacterial colonies along with the fungal filaments, they group and form a mesh-like structure called as flocks. Now these aerobic bacteria along with the fungus, they start eating the organic matter, or they start feeding on this organic matter, and when they do so, there is a decrease in the concentration of organic matter. As the number of bacteria increases, as the number of fungal filament increases, as they increase in number and start feeding on organic matter, it is a common understanding that even the requirement of oxygen decreases here. Here we have entered into a topic called as biochemical oxygen demand, or BOD, which means that the amount of oxygen which is required to feed up on the organic matter, or all the organic matter which is present in 1 liter of effluent and can be oxidizable by the bacteria. Now, as the bacteria feeds on the organic matter, now the organic matter starts decreasing. As the bacteria starts eating it, the bacterial colony will also increase, and the useful bacteria now started increasing. They started feeding on the different contents of the organic matter. That matter decreases now, which means that there was a requirement of oxygen, but now since the organic matter has started decreasing, even the oxygen requirement is also less. I can give you, or I can say you one thing, that the most polluted water contains higher amount of demand of oxygen, that is BOD, and as the polluted water started becoming purified and filtered out and become pure, the BOD also decreases. So BOD is high in polluted water and BOD is low in the fresh water. Since these flocks contain bacterial fungal filaments, started feeding on the organic matter, their size increases. It is a common thing. As the size increases with respect to the sedimentation process and settling process, with respect to their own weight and the gravity, they try to settle down. And this settled particles, now this is called as the secondary sludge or the activated sludge here, which once again we are not interested in. And the one which is freed from organic matter is allowed to enter into next process. Now we'll be taking a little amount of this particular activated sludge and we'll add it into the aeration tank. Remember the aeration tank? And we'll add it there just as a form of inoculum, just to make sure that the process continues as it is. Now remember, the one which was floating on top of it, which was resulted after the action of the flocks, that part is allowed to enter into anaerobic sludge digesters. This is also another tank, but we need to understand that this tank is completely anaerobic. Anaerobic means the lid is completely closed or it is not at all in contact with the air or oxygen. Here we see heterotrophic bacteria, those bacteria which are completely dependent on the other microorganisms for their growth and development. Common thing, right? Now in the digesters we have to understand that. Here anaerobic bacteria are there and heterotrophic bacteria are there, and these heterotrophic microbes start feeding on the useful aerobic bacteria and fungi. They try to eat them, kill them, digest them, and while doing so they produce different useful gases such as methane, carbon dioxide, hydrogen sulfide, etc., which will be further used in the form of biogas. Now dear students, this is the end of the topic, which is very easy to understand. As I mentioned, the topic name is the sewage treatment and the role of bacteria or role of microbes here. And here in this particular case we can see Methanobacterium as one of the anaerobic bacteria which completely destroys or digests different bacteria and fungal colonies and will produce methane, carbon dioxide, hydrogen sulfide, etc. A smaller topic which includes two stages called as primary and secondary treatments. Primary is the physical removal and secondary is the chemical or biological removal. So the actual use of microbes will be done in the secondary treatment or chemical treatment or biological treatment.
We are studying a chapter called as chapter number 10, Role of Microbes in Human Welfare, and today's topic is very small and yet easier topic. The topic name is Role of Microbes in Biogas Product. India is an agriculture based country, and to do that the Indians have got a lot of use of cattle. It may be with respect to milk, it may be with respect to draft work, etc. We use cattle in our day-to-day work, being a farmer of India. Now we need to think about one thing, that is the dung which is produced by the cattle. Can we throw it, or do we have to just think this as the waste product? No. IARI, Indian Agricultural Research Institute, and KUIC, Khadi and Village Industries Commissions of India developed a process. In this process or in this methodology, they have used the cow dung and they proposed a theory of production of the fuel such as methane from the gobar gas or biogas. Now, the production of methane as a fuel takes place in two types of situations. One, the name is called as biogas, and two, the name is called as gobar gas. In both the categories, there is a production of methane. In case of the biogas, the raw material can be anything, which can be biodegradable. But in case of gobar gas, the raw material should be cow dung. So, what is a biogas? Biogas is nothing but a mixture of inflammable gases such as methane, sulfur dioxide, carbon dioxide, etc., which are produced by the microbial activity and can be used as a fuel content. The cow dung contains a cellulose as the material, and this cellulosic breakdown should be done by one of the bacteria called as methanogens or methane producing bacteria. These methane producing bacteria or methanogens work only in the absence of oxygen, and they anaerobically break down this cellulosic material and produce methane, hydrogen sulfide, carbon dioxide, etc., and can be used as a fuel for our future purposes. Rumen, a part of the stomach of the cattle, wherein which these methanobacterium are present and provide nutrition to the cattle. Now let us go to the simplest procedure of how actually the methane gas or gobar gas is produced. Now let us go to the diagram. In the diagram, we have got three tanks. Let us name them as tank 1, tank 2 and tank 3. Let us dig some holes now. Now for the tank 1, around 3 to 4 feet, and we'll add two things, one as cow dung and another one as the water. Equal proportions of cow dung and water are mixed up in the tank number 1. Let us go to tank number 2. This is a longest one. We have to dig around 10 to 12 feet or 10 to 15 feet in the ground, and it is a large container. And the third one is wherein which the outlet sludge or waste product will be collected. As I mentioned, in the tank number 1, we need to add cow dung and the water. We need to mix it, agitate it. This mixture is allowed to pass or enter into the tank number 2. Now, the mixture of cow dung and water enters into tank number 2. There is a lid or gas holder which is present on top of this tank number 2. Now this is not at all completely fixed, it is just floating on top of it. As the methanogen starts working on the cellulosic material, it starts producing the gas. These gases such as methane and carbon dioxide and hydrogen sulfide, etc., they push up the gas holder or the lid which is floating on top of it. There are two types of outlets. Outlet number 1, which contains the useful gases, is allowed to enter into the household activities as a fuel, and the outlet number 2 is the one wherein which the waste slurry or the waste product or the waste dung material which are already been used will be kept. And this particular waste in the future is used as the manure in the agricultural practices. Very simple. The biogas can be used as a fuel for cooking and lighting purposes. This is it. Dear students, this is the end of the topic, and I hope this is very simplistic and very easiest topic in this chapter.
Today we have reached one of the topic called as the microbes as biocontrol agents from the chapter called as the role of microbes in human welfare. This is also one of the easiest topic to understand. First of all, what is the meaning of biocontrol? Biological control or biocontrol means that it is a method of controlling pests such as insects, mites, weeds, plant diseases using other organisms. Let us clear this sentence. Imagine there is a plant. The plant has got leaves, fruits, root, stem as of the major eatable products for one of the organism. Consider that one of the organism as enemy A. Now we want to remove, eradicate, delete, kill this enemy A, but without the use of chemical fertilizers or pesticides or weedicides. From the natural studies by the different agricultural practitioners and the botanists, we come to a point that this enemy A has got another of his own enemy B. This enemy B's major food is enemy A. Now allowing this enemy B to enter into the farmland and make it to hunt enemy A so that the concentration or the number or the population of enemy A decreases. We know a fact that enemy's enemy is a friend, right? So by understanding the same concept in our minds, that it is the removal of the weeds, insects, pests by using other organisms as the definition itself understands. Before the use of biocontrol agents or the use of biocontrol, there was the use of insecticides and pesticides. The farmers have started rejecting and started less use of these insecticides and pesticides and weedicides. Now here are some of the points why the farmers have rejected this. Point number 1. The chemicals used in the agriculture are harmful. They are toxic to the humankind and even to the animals. Point number 2. The overdose use of these chemicals may harm the health conditions of animals and even the plants. These can even pollute the environment, even with respect to plants also. Point number 3. We use weedicides for the purpose of removal of weeds, but this can even result in the pollution of upper layer of the soil. Point number 4. The organic farmers with their techniques of removal of these insects, weeds, mites, etc. are of only the temporary purposes. They don't use this for the complete eradication of these pests. After understanding why actually the use of pesticides and insecticides got cancelled, let us enter into studying of some of the basic examples of biocontrol agents which are given in our NCERT syllabus. One of the major enemies of the natural growth of the plants in agriculture are aphids and mosquitoes. For the removal of aphids, we use ladybirds as biocontrol agents because the ladybirds have a natural affinity towards aphids. They eat it and continue their life cycle. it is come to one understanding that the removal of one enemy called as aphid is done by its own enemy called as the ladybird. So the ladybird comes and eats the aphids so that the removal or the eradication of aphids takes place. The another thing is the removal of mosquitoes is done by the dragonflies. The same explanation goes with respect to ladybirds also. There is a bacteria called as Bacillus thuringiensis. It produces toxin, and that toxin is highly dangerous for the growth of caterpillars of butterfly. These Bt toxin are used to kill or to destroy or to decrease the concentration of butterfly caterpillars. Even the dried powder of the Bt toxin mixed with the water and sprayed onto some of the plants such as brassicas, etc. When the insects feed on this particular toxin, it enters into their gut and it becomes active and releases the toxin and kills the insects. Nowadays, even this Bt toxin have been genetically engineered and has started using in the production of genetically modified plants. I can give you an example called as Bt cotton, Bt brinjal, Bt tomato, etc. etc. Some plant pathogens infect the root system of several plants. In this case, we use Trichoderma species as the perfect answers for the complete eradication of these plant pathogens which infect the root sections of several plants. We go to another example. Baculoviruses are a kind of pathogens which are used to control or kill or to destroy some insects and arthropods. Majority of the biocontrol agents belong to a genus called as nucleopolyhedrovirus. These are species-specific and narrow-spectrum insecticides. Insecticides are the chemicals or the one which kills the insects. Baculoviruses are even used in the IPM section, that is called as Integrated Pest Management System, which is even beneficial for the farmers to do their agricultural work. Dear students, I told you or I explained you in a very short manner about one of the easiest topic of this chapter called as the role of microbes as biocontrol agents. In this topic, I did not completely have given 100% examples of only the microbes. I have used some of the insects and prey-predator mechanisms. The explanation of this topic is only one thing: using the other organisms to kill the pathogens or the insects or the mites or the mosquitoes which are dangerous for the plants to grow and even dangerous for the farmer to do his work in the agricultural sector. I hope you understood this small video.
Today, we'll be studying the last topic of the chapter called as Microbes in Human Welfare, and the topic name is the role of microbes as biofertilizers. Dear students, in case of agriculture, whenever a farmer encounters a plant with a different disease, he tries to apply different kinds of chemicals onto them, such as pesticides. These are the chemical agents which can kill or remove the pests. Weedicides. These are the chemical agents which can kill or remove the weeds. Bactericides. These are the chemical agents which can kill the bacteria. Viricides. The chemical agents which can kill the viruses. Insecticides. The chemical agents can kill the insects. Fungicides. The chemical agents which can kill the fungus and their related genera. Now the point here is only one thing. The farmer felt that there is a disease which is causing the plant to die. That's why he is using all these different chemical agents. Of course, the disease will go away. There is no secondary thought of it. But we need to understand one thing, that the regular overdose exposure of these chemicals might lead to the death of the fertility content of the soil.
death of the quality content of the product, death of the quantity content of the product, which will be released by the plant and of course from the agriculture itself. Now the scientists, botanists, agriculture practitioners thought of secondary ideas here. One of the idea is using the biofertilizers. Now, what is the meaning of biofertilizers? These are the microorganisms which enrich the nutrient content of the soil, the fertility content of the soil. Whenever I say the nutrient or fertility content of the soil, it should have to have these kind of basic ingredients in them, such as nitrogen, phosphorus, potassium, etc., etc. And it should be done by the microorganisms only. That's why the topic, the role of microbes as biofertilizers. In this topic of biofertilizers, we'll be concentrating on three different categories of microorganisms, such as 1. Bacteria, 2. Fungi, and 3. Cyanobacteria. We'll be studying all these things detail by detail, one by one. So let us study the role of bacteria as a biofertilizer. Bacteria number 1, Rhizobium. It is a symbiotic bacteria which lives in the root nodules of the legumes and helps in fixation of nitrogen and converting that into the organic compounds. Symbiotic bacteria is that kind of a bacteria which lives in the mutual associationship with itself to the plant roots, Azospirillum and Azotobacter. These are the free-living bacteria which absorb free nitrogen from the soil, air, and convert them into the salts of the nitrogen, such as amino acids, and these enrich the soil with its nutrient contents. Dear students, remember I told you about the bacteria as biofertilizer and I have mentioned three different examples. The neat student should have to remember here is that the Rhizobium is a symbiotic bacteria and Azospirillum and Azotobacter are the free living bacteria. Let us go to the point number 2 called as fungi as biofertilizer. Fungi forms the mutualistic relationships or symbiotic relationships with the roots of higher plants called as mycorrhiza. I can give you an example called as glomus. The fungal network called as hyphae absorbs phosphorus from the soil and it transports that phosphorus to the different parts of the plant, wherein which in return it takes only the shelter in the root nodules of that plant. Let us study some of the benefits of mycorrhiza. Since it lives in the roots of the higher plants, of course it gives a resistance to root-borne pathogens. In case of salinity conditions, that is high salt concentration conditions, and drought conditions, which means that highest temperature and lowest water level conditions, also it gives us a tolerance against these kind of situations. Mycorrhiza even helps in overall growth and development of the plant. Okay students, let us enter into the third category of this topic, and that is cyanobacteria as biofertilizers. They fix the atmospheric nitrogen and they fix the nitrogen content and the fertility content of the soil through their photosynthetic activity. Some examples include nostoc, anabina, oscillatoria, etc., etc. Cyanobacteria or blue-green algae also increases the fertility of the soil by increasing the organic matter content into the soil. That's it. This is about the role of microbes as the biofertilizers, and I have given only the content which is present in our NCERT syllabus with respect to graphical representations. That's it.